So, welcome everybody to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we're really happy you made the time to dial in today. And we're here talking about a tale of two selectivities with Danette Daniels today. So again, Danette, it's a pleasure to have you, and really we're honored that you're here with us today.
Yeah, great. Thank you for having me. And thank you to everyone who tuned in to listen to the story today.
And yeah, without further ado, I will actually introduce our guest, Danette. Danette serves currently as the vice president of the protein degrader platform at Foghorn Therapeutics, where she's developing therapeutic degraders in the areas of chromatin regulation and oncology. She was a very early leader in the field when nobody was talking about targeted protein degradation, and she was pioneering approaches to monitor cellular kinetics of degradation, understanding the mechanism of action, and co-developing new PROTAC modalities. She's a member of numerous committees, including the AACR Chemistry and Cancer Research Steering Committee, the HESI TPD 2026 AACR Chemistry Working Group, and was just co-chairing, I should say, the 2025 AACR New Drugs on the Horizon sessions. She will be leading a very exciting new Gordon Research Conference series on induced proximity modalities and therapeutics that is set to begin in June next year. So you should already kind of look out for that. That'll be very, very exciting. In February of 2023, Danette actually co-founded the Women in TPD and Induced Proximity Initiative, which is nearing 1,000 members and offers mentorship programs in addition to conference travel scholarships to early career scientists. So a very important initiative here as well. Prior to Foghorn Therapeutics, Danette received her PhD in biophysics at Yale University and was a postdoctoral fellow at the Stanford School of Medicine, where she was studying the Wnt signaling pathway. Before joining Foghorn, she was a group leader at Promega. There she was involved into revolutionary technologies in the assay field, such as halotags, nanolukes, high bits, and the nanobread technology as well. So Danette, really impressive CV. Welcome to the show. Again, we're honored to have you today.
Thank you so much.
Yeah, and in the good old Drug Hunter Flash Talk tradition, we're starting this with a few questions to learn more about you as a person. So I'd love to hear more about what inspired you to start a career in drug discovery.
Yeah, so I've had a long career in the biotech space, as you mentioned, in technology development, as well as some more basic or academic scientific research. And in my previous role at Promega, we did a lot of collaborations with people at pharma companies or small biotech doing drug discovery. And I was always involved in those early stages of the project, like trying to figure out the mechanism with them, trying to figure out ways to better triage the compounds. But I always had this question in my mind of, I wonder why they chose this target. What indications are they thinking about this target? What's going to happen to this program after we finish this initial analysis? So that kind of like real world practical translation experience was really something that I was driven to do. And when I had this opportunity 3 years ago to come to Foghorn Therapeutics, not only was it in the field I loved, chromatin biology, but it was also in degraders. It was kind of like the perfect alignment of things that I was looking for, and an opportunity to really understand the process and be involved then in development of drugs, to oncology, which I cared so much about and studied for so many years. So that translational opportunity and potential impact we can have in patients' lives really, really drove that decision.
Awesome. Yeah, no, that's super, super inspiring. Yeah, Danette, as you mentioned, you've worked on many, many targets, and I'm sure you have a favorite molecule. Can you talk a little bit more about your favorite molecule and why it is your favorite molecule?
Yeah, so when I first thought about this, in the back of my mind, I'm a parent. My favorite molecule was children's ibuprofen. It got me through many
I love
sleepless nights. But if I put on my scientist hat, I was just a co-chair of the New Drugs on Horizon. And there were fantastic talks from Brian Landman about the new Amgen KRAS inhibitor and Ann Edwards about revolution medicine. And these disclosures really embody just the possibilities that you can have of new structures, these larger molecules we wouldn't have thought would be feasible many years before. But honestly, my favorite molecule is the one we're working on now. I mean, it's the first drug discovery molecule and degrader for chromatin targets that I've ever been very personally involved with. We don't have the structure disclosed yet, but the CBP degrader that utilizes VHL is really an outstanding structure. And I think when we are able to disclose that structure, it will be very striking and impressionable for the field.
Awesome. Yeah, we definitely look forward to that. And yeah, you're definitely bringing up a lot of great points there as well. And what are you excited most about in the field today in drug discovery in general?
Yeah, well, my focus really for the past 20-something years has been on chromatin and epigenetics, and then transitioning from inhibitors to degraders. But you see so much growth and innovation in thinking about new modalities, new ways to regulate biology, new ways to drug targets. And I think what we saw in that transition from inhibitors to degraders, where we had inhibitors that just didn't have efficacy. They had fabulous binding and selectivity of binding, but lacking the efficacy. And so we went the step further to degradation, and now we have the efficacy. And this induced proximity concept that degraders are a part of, we see now just unfolding in all sorts of different directions, from protein stabilization to relocalization to rewiring of transcription. And I think, degraders were really a seed of an idea of what was possible when you start to think beyond, your one small molecule to your one target, right? What now can we bring together where maybe the outcome is not degradation, but the outcome really opens the door for the biology that we now have the potential to modulate and use then to treat disease.
Yeah, no, I love that. Yeah, that's a super inspirational. And earlier in the pre-discussion, we were talking about trams and all these
Yes.
cool concepts. So yeah, really excited for in general, the induced proximity field. And, the degraders have really opened the door to a whole new field of drug discovery. Super
Yes.
excited about that myself too. If you could give yourself some advice, your younger self some advice, or maybe younger people in the audience some advice, what would you say to your younger self? What would you maybe do different or do the same that in your career?
Patience. Really. Yeah, we talked about it earlier, when the initial days of degraders, there was a lot of pushback. What are these, 1,000 Dalton compounds? These will never be drugs, but they were giving us the biology that we really needed to treat disease. And so it's a lot of work really to not be constrained by the rules that we had before, and find a way. How do we make these more drug-like molecules, and just staying with that throughout all of these years, and seeing the evolution of it and all of this work that has been put in by many, many people, Foghorn included. They were one of the first companies to take a degrader into clinic. You know, it just really told you, don't give up, even on these big ideas, right? That might seem impossible at first. There's really opportunities there.
Love that. Yeah, I definitely have to take some of your advice. I think I'm one of the most impatient persons.
As am I, Dennis.
Awesome. Awesome. Danette,
Yes.
one more question before we get started. What do you like most about Drug Hunter?
Oh, Drug Hunter has something for everyone. I'm not a medicinal chemist, right? I love chemistry. I love to look at all the trends and such. But I'm a trained structural biologist, and my team does a lot of cell biology and mechanism work. But, you know, it's so nice to have something that puts and links the structural elements and highlights the novel features and the advances that have been made on the medchem side directly into how this impacts, you know, potential disease indications, where it could be useful, and really ties that full story that you need in drug discovery, from the innovation on the chemistry side to the enablement of the biology, and ultimately the impact this has for new treatments.
Yeah, no, that's great to hear that, you know, it's useful beyond medicinal chemistry.
Yes.
And that's amazing. And yeah, I want to share like a few things that you mentioned. I want to show people on the Drug Hunter website real quick. So one thing you mentioned is the AACR, that's the session you co-chaired, right? Here, the AACR
Yeah.
Drugs on the Horizon session, and people can actually find all the structures that are being disclosed at that meeting here in this one single poster. And we're starting to put together the case studies for these as well that you will be able to read on Drug Hunter shortly. One thing that I did want to show that we just recently introduced is our new tab here with recent disclosures. So if you click here in our recent disclosure database, you'll be transferred to that new database. And here you can search for targets. You mentioned KRAS, so you can search for example, KRAS inhibitors or KRAS degraders. You can search by a therapeutic area. So if you're interested in what's new in the oncology field, you can just type in oncology, see a few new targets that people are working on that are recently being disclosed in the literature, preclinical or in clinical phases. You can type in a company, for example, let's say AstraZeneca. You can see what is AstraZeneca currently working on, what kind of stuff do they have ongoing that's exciting in the literature. And again, you can also type in a phase of drug discovery clinical research. For example, if you want to see assets that are being disclosed that are in phase 3, you can just type in phase 3 and get a list here of assets that are currently in phase 3. So for example, if you wanted to learn more about this TYK2 inhibitor here, we actually just had a brief on this. So you can see here's this TYK2 inhibitor with biologic-like efficacy here from Alamis in San Francisco. And you can read this little highlight, this little profile of the molecule here on Drug Hunter as well. And with that, I will stop sharing my screen, and I'm super excited for Danette's talk about the tale of two selectivities, and excited to learn more about CBP and EP300 as super cool epigenetic targets that we can degrade rather than inhibit. Danette, floor is
Yeah.
yours.
Great. Okay. Yes, so thank you again, everyone. I'm really excited today to talk about our two preclinical programs of CBP and EP300 degraders, and really an in-depth analysis of achieving selective degradation and our understanding on how we were able to do this. And then, of course, the applications now that we have these selective degraders in hand that we can use within oncology. So as I mentioned, I've been in chromatin and epigenetic and transcriptional biology now for almost 20 years. And in my world, all roads lead to chromatin. And really, if you think about the action of many drugs, from, say, blockers of cytokines to GPCR, EGFR, all the way through kinase inhibitors or agonists of nuclear receptors, that end signal really is regulation of chromatin and regulation of transcription in order to control the disease. And oftentimes in oncology, what we see are drivers of proliferation, so unregulated cell growth, aberrantly on. And it was really such a privilege to come to Foghorn. They have this deep and rich understanding of chromatin binding proteins, the structures that regulate accessibility to chromatin, the understanding of this toggle and nuanced switch between activation and repression, and how transcription factors can be brought in to recruited to really amplify these signals. And what we see is that many of these chromatin regulatory proteins are implicated, particularly in oncology across a wide range of cancers. And this really opens the door for multiple opportunities of thinking about targets to drug. And we see, especially in chromatin, that several of these complexes, particularly listed here is the chromatin remodeling complex, BAF, which was really a foundation for Foghorn with the pioneering work of our co-founders, Drs. Sigal Kadosh and Jerry Crabtree. But we see within this complex, there are paralog relationships. You can have cancers with mutations in one of those paralogs, and this really opens the door for taking advantage of synthetic lethality, as well as you'll see in for a CBP and EP300 proteins, lineage dependencies. So Foghorn works on these factors. One theme that we have really pressed upon and been successful at addressing with these factors is achieving selectivity. And this has been historically difficult for many in the field that have worked on these targets. We are the first to have a selective SMARCA2 inhibitor in clinic in partnership with Lilly. And this really, this is a paralog situation with SMARCA4, and we know that SMARCA4 is mutated in a significant number of cancers. We also, I won't talk about it today, have an ARID1B program, a degrader program, and we disclosed at the start of this year that we have achieved selective ARID1B degradation. And this also is a synthetic lethal relationship with ARID1A, but ARID1B is a very, very special program. It was started at the beginning of Foghorn, and it's one of these targets which is not an enzyme, and it didn't have any known binders or even any domain within it that had been previously drugged. So really an excellent example of success in applying degradation and drugging a historically intractable target. So I'll spend the rest of the time talking about CBP and EP300. These also are paralogs. And while unlike ARID, they had been drugged before, but the difficulty with those inhibitors to CBP and EP300 was that they were not selective. They had dual activity against both. So CBP and EP300, these are key acetyl transferases, and they acetylate histones as well as many proteins. And this mark of acetylation will typically result in activation of transcription. And I mentioned before that dual targeting, while it has been achieved, has revealed tolerability and safety issues, and this has been demonstrated as well in clinic. So we see that these proteins, they have been on the radar for many, many companies over the past several decades. because we do see dysregulation of these in many cancers, and we see some cancers have dependency on one or the other. And then also we see many cancers that have one of these mutated or loss of function. So we have both synthetic lethal opportunities as well as lineage dependencies. So what has been the challenge of these two highly related proteins is not that they don't have domains that accommodate small molecule inhibitors. It's that these domains have high sequence identity. And no one has been able to develop either selective histone acetyltransferase inhibitors or selective bromodomain inhibitor as well. So we thought, okay, this is a perfect situation to develop a degrader. And in that degrader, can we develop selectivity? And that's really because degradation is more than occupancy. We refer to it as event-driven pharmacology. Indeed, you have an occupancy step of your drug binding either an E3 or the E3 and the target. It depends on whether it's a molecular glue or a PROTAC. But really, that occupancy is just the first step. You then have a second step of inducing the interactions. And while you may be able to have success at the induced interactions, what you really want are these positioned well for your functional outcome. And that event in this case will be degradation. So we have this kind of simple thought about degraders. You know, our entire goal using this modality for drug targeting is to really override the native homeostasis that keeps your present or your target present. And so we want to apply the degrader to really drive down clearance of the population, but then we need to sustain that clearance. And because it's not that the protein is gone, the cell is trying to resynthesize that protein. And for cases in cancer where this might be a disease driver, the cell is addicted to this protein, there might even be feedback loops that tell the cell to make more of it once they sense they have lost the levels. And so this really puts into a picture more of not just this simple thing like we have an endpoint of degradation, but we really have to keep the pressure on these targets to keep the target down and prevent this recovery from occurring. So there's many components. I'll talk about some of these mechanisms where you can influence that efficiency of degradation, so the speed of degradation. But also you can influence how long the target stays down before maybe you have to redose. And clearly compound stability, especially in the context of heterobifunctionals, efflux, all of these factors will play into how long can we keep with any concentration our target degraded. And lastly, because we use machinery to co-opt the process, we have to have that machinery around. And so any type of variability in those components that we're co-opting, or maybe even changes in endogenous target protein level, can really influence your success in amount of degradation, as well as that time-dependent sustained degradation. So we started with a bromodomain binder of CBP and EP300. Again, this is a dual bromodomain binder. We then linked it to several different E3 ligases. Here I'm showing you a linkage to the VHL E3 ligase. And we saw immediately, we got excellent dual degradation. So CBP down, EP300 down. And these are high-bit cellular assays. The CBP and EP300 are endogenously tagged. And these are live cells. So you're adding the compound to this cell and you're watching how fast does it be depleted, and then how long does it stay depleted. And you can see this dual degraders really kind of set the benchmark for us, saying how quickly we could clear the population and to what level could we clear that population. So we have complete loss here, Dmax in the fast rates, and you can see over a significant concentration range, we're able to maintain that loss. But dual is not what we wanted. We wanted selective. So I'm skipping through thousands of compounds made and tested, and I'm showing you selective degradation. This is exactly what we want. We're adding a compound and we're only impacting the levels of CBP. You can see we have even faster rates here for CBP than we had on the dual. We see complete flattening of Dmax across this concentration range, and over that same concentration range, even up into the micromolar, we see that we're not degrading the EP300. So this actually is a picomolar degrader. In parallel, we're managing separately the EP300 program, and we're very pleased to see that with an EP300 selective, we can flip the switch. So now we're only seeing loss of EP300 without impacting the levels of CBP. So this is great. We're looking at just these two proteins. What is super critical in degraders is to do global proteomics. What else potentially could we be impacting? The answer here is CBP and or EP300 are the primary targets that are being lost. We looked in cell lines that were not dependent on either CBP or EP300 for survival and contained both CBP and EP300. You can see over here on the very left-hand side with a CBP selective in a U2OS cell, which has both proteins, we only see loss of the CBP in global proteomics. We additionally looked, because this is a bromodomain binder, are we seeing any loss of other bromodomain proteins? The answer is no. And then we can see also selective CBP loss in a cell line, RKO, which lacks EP300. And then similarly on the EP300 side, we see this excellent selectivity recapitulated, but now for EP300. So this is fantastic. We've achieved selective degradation, but we started with a dual inhibitor. So yes, we are degrading in the picomolar, and the dual inhibitor is in the nanomolar IC50. But our question really was, could we make that window wider? And now you're really flexing the power of degraders, because to get excellent degradation, you do not need high affinity binary engagement with your target. This again is decoupling that occupancy step with then the induced proximity of the ternary complex and the functional outcome. So now we took on an active campaign that you would not take on in an inhibitor, but we weakened the inhibitory binding basically to the off-target here for a CBP selective degrader, the off-target inhibition on EP300, and yet still pushed to get high potency CBP degradation. So you can see, yes, there is a trending. If you weaken the affinity for your target, you do trend with more compounds over in the lower degradation potency, but we were able to find significant candidates now that had weaker IC50s on EP300 for inhibition and very, very potent degradation. So we now have a selective degrader that has minimal dual bromodomain inhibition. And likewise, we could do this on the other side with the EP300. So very pleased now to have these differential compounds where we're really separating the degradation event from the inhibitory aspect within the compound. So mechanism, we get asked a ton, how did we achieve this? I showed you on the previous slide, tons of compounds. I mean, fantastic med chem effort combined with rational design and empirical design and computational chemistry. But really, once we got very, very selective compounds, we wanted to also look to see how ternary complex was affected by having selective degradation. So here we have the VHL with the Langen BC, or what we refer to in the field as the VCB complex. with the degrader and the CBP bromodomain. And we could very nicely see the interface between the CBP in green and the VHL in blue with the CBP selective. We then crystallized and solved the structure of the EP300. And we saw that it adopted a different conformation, and it actually formed a slightly different interface with the VHL. So you ask, what's going on in the dual? Well, actually, the dual adopts yet another third conformation. And this now engages different residues also within VHL. So it's really this beautiful example of moving the ternary complex docking in this highly, I mentioned there was 97% sequence identity of the bromodomain. But this really shows you how in the design of your degrader, you can really change the docking with your E3 ligase. So we wanted to do follow-up studies, biochemical, to look at affinity and cooperativity and favorability in the ternary complex. So we moved to TR-FRET, and this now allowed us, we first looked at the bromodomain degrader interactions. So we have CBP, IC50, or EP300 IC50 on the top with a dual degrader, and on the bottom with a CBP selective. You can see that EP300 line has shifted here between those two. And then we looked at the VHL engagement, and you can see even by this plot in this binary analysis, we have very tight engagement on VHL, especially with the CBP selective degrader. We then moved to SPR to look at off rates, and we find that it is quite stable engagement and slow off rates on VHL with both the dual degraders as well as the CBP selective. So now what about the ternary complex? We then turned to SPR. We looked in the dual degrader and we added the CBP bromodomain or the EP300 bromodomain, and we saw that these were quite stable ternary complex, so slow off rates. Now, when we looked at the CBP selective, you can see right away that we have that same stability on the CBP ternary complex, but now our EP300 off rates are very fast. So we haven't prevented ternary complex formation with our anti-target, the EP300, but it is far less stable in terms of off rates, so staying intact as a ternary complex. Now, when we put this together with the structural studies, we think about now positioning in the larger colon ring 2 ligase complex, and this is a model based upon the fantastic work done by Brenda Schulman in the cryo-EM determination structure of the full CRL2 with the E2 and the ubiquitin poised, ready to transfer ubiquitin to the target. But we've aligned here into that structure based upon VHL. And you can see immediately, even with just the segmented bromodomain, we have significantly different positioning within this larger complex that would impact that distance or the lysine availability of those to ubiquitin, to those lysines. So we believe there are many factors here at play. First is the docking onto the VHL. Then there is the stability of that docked ternary complex on VHL, and this is all translating to changes in target ubiquitination. And for time, I did not include, but we have cellular data to show that the anti-target is just not ubiquitinated inside cell. So this really is a broader mechanistic study, and we're really delighted to see the differences with these selective degraders. So now I want to move, now that we have these degraders in hand, what can we do with them? What potential indications can we address? And so we move to, first with our CBP selective degrader, to looking in vivo, in xenograft models now, tumor growth inhibition in a variety of cancers that had EP300 loss. So this is taking advantage now of that synthetic lethal relationship, but you can see we have very nice TGI across multiple of these models. And you can see in gastric cancer, we actually see regression at several of the concentrations here, all with minimal impact on body weight. But we started this, it's fantastic to see this TGI. What we were really hoping in design of a selective compound was getting over some of the tolerability challenges that dual even bromodomain inhibitors see. And really that has been thrombocytopenia for these two targets. So we then immediately looked in mice whether or not we are having an impact on the platelets. And indeed, we see with a selective CBP degrader, and these are shown at 2 different doses, 3 mg per kg, sub-Q daily in the blue, we do not see that loss of platelets. And as controls, we put in a dual degrader in green and dual inhibitors in the pink, orange, and purple. And you can see all of these. As soon as you have some type of dual activity on both CBP and EP300, you see loss of the platelets. A nice study was published by Genentech looking at rat models, looking at the staining of bone marrow and the megakaryocyte population. So with their dual bromodomain inhibitor, GNE781, they did see loss of megakaryocytes, which are the precursors to platelets. And we now, in our investigational study with our selective CBP degrader, do not see that impact either on the megakaryocyte population, meaning that from even the precursor, we are preserving the platelets. So with this selective degradation, we have improved that safety and tolerability window. So I mentioned it's VHL. Anybody who works on degraders knows how incredibly difficult formulation is for VHL. So we've had significant breakthroughs here at Foghorn in thinking about formulation, especially for VHL-based degraders. And this has been our development of our long-acting injectable platform. And beautiful work, and I'm highlighting the big breakthrough here in the formulation and being able to deliver this, but we now compared a single dose LAI injection, and we looked at TGI again in this EP300 loss model, and you can see this beautiful regression that we have with a single LAI dose over the course of 3 weeks. I have the PK/PD shown for both the LAI, and then we compared this to a daily dose of 10 mg per kg sub-Q of the same compound. And we actually see improved PD and a much higher sustained PK when we do the LAI. So this is really remarkable. And you might be thinking, oh, CBP, maybe it doesn't have a fast turnover, or it has a really long half-life. It actually has a half-life of around 10 hours. So it's really, really impressive to hold the target down for this length of time with this single dose. And again, no thrombocytopenia was observed at the end of this study. So we figured out, we've looked at TGI in these synthetic lethal populations. We also wanted to explore what about possible cell lines that had some CBP dependency. And one of the lines that stood out to us in our PRISM screening and analysis was ER-positive breast cancer. And this is a cell line that has both EP300 and CBP, yet the two seem to play different roles, and we're really seeing this dependency more so on CBP. And I have here MCF7, and these are colony formation assays. So we're looking at impact on cell growth or cell defects. And you can see already a single agent, our CBP selective degrader, definitely impacts, even a single agent, the growth of these. And so we wanted to look at combination with standard of care agents for ER breast cancer. What I have here highlighted in green is that combination with just fulvestrant. And already you can see we have synergy in that binary combination. And then when we do the triple, when we look at the CDK4/6 inhibitors, we see also just in that dual combination with the CBP degrader synergy as well. And we see even further improved synergy when we do the triple combination. We looked not only at MCF7, but we also looked at T47D. And I think you can appreciate that we also see, again, single agent activity, but improved activity for impeding cell growth with either fulvestrant in a binary combo, or the CDK4/6, or in the triple combination. We're really investigating now this further and moving in vivo studies, but we were really intrigued by this combination with the CDK4/6 inhibitors, especially given the impact on, say, cell cycle control, as well as the transcriptional regulation that our CBP has. So we went back and looked in our EP300 null lines to see if there was synergy just in general of our CBP degrader with the CDK4/6 inhibitors. And indeed, we saw, especially at these lower concentrations, excellent synergy across many of these cancers that are EP300 null with the CDK4/6 inhibitors. So we're going to be investigating this further, as well as combinations with standard chemotherapy agents as we prepare for clinic. So that's the CBP selective side. I'd now like to switch gears to the EP300 selective side. And when we look at dependencies, we don't see a lot of cell lines that have CBP mutations. So a minimal number maybe for that synthetic lethal relationship. But what we strikingly see with EP300 is lineage dependency, specifically on EP300 and not CBP. And the two that we focused on are the heme malignancies, as well as AR prostate cancer. So now we have the perfect tools, really, to get after the biology. A lot of the field thought that EP300 and CBP were just redundant. They would step in for each other. But now that we have a CBP selective degrader and an EP300 selective degrader, we could really drive down and figure out which one of the two is regulating the signaling. So in prostate, we looked at all these AR genes, so AR, the variant ARV7, and MYC, and other related genes, and we could treat with a CBP selective degrader, and we didn't really see any modification of these genes. But when we treat with the EP300 selective degrader, now we see the downregulation of these AR signaling genes, and that EP300 degrader mimics or looks identical to when you treat with a dual degrader. So this really indicated to us that EP300 is the driver. We know it works in concert with AR to keep the AR genes active. And we see now, again, in colony formation assays, this significant impact on growth with the EP300 selective degrader. When we make the ligase dead version, so we've got still that inhibitor on, much less so, but the EP300 degrader mimics the dual degrader. We then moved into xenograft models, into a VCAP prostate. We saw a very nice dose response and TGI, significant TGI in the VCAP model with our EP300 selective degrader. What is shown here in the Western blot is an end of study tumor lysate, and you can see that selectivity playing out even after the end of this study, and minimal impact on body weight change. So what about in resistance setting? This is a big population within prostate cancer. So we were able to obtain an enzalutamide resistant population VCAP model. You can see here the treatment with enza in this model no longer impacts the AR target genes. And but very nicely, we could see that treatment with the EP300 degrader now could impact the cell growth. So we have an orthogonal approach for still modulating the AR target genes, but through EP300 loss, rather than relying upon modulation of AR itself, even when you've become resistant to the AR targeted therapies. So this was prostate. I want to switch gears now to the heme malignancies, and this is multiple myeloma. We found that the vast majority of multiple myeloma lines are sensitive across varying concentration ranges to the EP300 degradation. This is anti-proliferative effects. And then Western blots showing, again, this selective degradation that we see. These cell lines, many of them do express functional full-length CBP. So again, we see the separation of the two proteins and their impacts on cell growth. We also find that DLBCL in general as a class is very, very sensitive to EP300 degradation. And here is just a comparison of our degrader in dark blue with a ligase-dead version. So that now has that inhibitor aspect. And when you ask the question of what does degradation buy you versus inhibition, this is a great example of that. So with the inhibitor, yes, you see some of the lines are sensitive, but by far you get much greater impacts now with degrading EP300 rather than inhibiting it. We then moved into a in vivo model in DLBCL in the CAR plus 422. Again, we see significant TGI with the degrader in this model. Again, end of study, loss of EP300 with minimal impact on body weight loss. This is with the VHL-based degrader. So we went then into look at combination with standard of care agents for both DLBCL as well as multiple myeloma. So for DLBCL, we looked at combination with increasing doses of dexamethasone. With multiple myeloma, I'm just showing here pomalidomide, but we also looked at lenalidomide as well as dexamethasone. And we see significant synergy of the EP300 degrader with both of these standard of care agents in DLBCL as well as multiple myeloma. So the big thing in multiple myeloma is that imide resistance can emerge either from prior treatment, or there are just some multiple myeloma cell lines which are not that sensitive to the imides. And so here we wanted to look to see, again, very similar in context to the enzalutamide resistance that I just showed you. Does EP300 offer an orthogonal strategy for anti-proliferation than these other agents? And now here you see in the green hexagons and the dark green, these are many lines in this panel completely resistant either to pomalidomide or lenalidomide, but we see very nice cell killing with the EP300 degrader in these lines. And we are doing more investigative studies on this. So this is VHL-based degraders. We also wanted to get a cereblon-based degrader. Those, again, of you that work in the field know that cereblon, oftentimes in a better bifunctional, can be more amenable to oral bioavailability and delivery in this fashion. And we were very pleased. Again, I'm glossing over so much chemistry and so many compounds tested, but just the team was fantastic to find now selective degrader with a cereblon base, and we're not impacting the CBP levels. they were able to formulate this for oral delivery, and we went back to the multiple myeloma and did the study comparing our EP300 degrader and the MM1S, a very sensitive line, and we see excellent TGI. We did compare against the current phase 1 clinical compound, which is enobardiv, a dual bromodomain and dual CBP EP300 bromodomain inhibitor, and we're seeing comparable TGI. However, when we go to look at the platelets, we do see that reported impact that the dual inhibition has reported out from clinic on the platelets, but we don't see it with our selective EP300 cereblon-based degrader. So with this, I want to summarize this tale of two selectivities. We've actually been able to take it in both pathways, from CBP selective to EP300 selective. And we find that having these two programs really allows us to think about a lot of potential indications, a lot of exciting combinations that we can have to really reach patients that might be resistant to current therapies in these various indications, and allow a different mechanism to really get at slowing down or killing those cancerous cells. And we've also really made breakthroughs, I think, in the formulation, particularly on the VHL side with the long-acting injectables. These are big learnings and for the entire field, where the majority of clinical compounds are cereblon based, but we'd really like to open up this space as well. So, and with all of these developments on both programs, we see that improved safety window and tolerability in the thrombocytopenia, protection of the megakaryocytes, as well as the platelets. And with that, I would really like to thank the Foghorn team. I'm really just the person today that gets to share the exciting work of so many teams. It's just a wonderful place to work, so inspiring. And I feel very fortunate to work with all the teams, and their names are listed here that worked on both of these programs. And before I finish, because Dennis mentioned, we started 2 years ago this Women in TPD and Induced Proximity initiative to really highlight the accomplishments and bolster up the community and provide a networking support for the community. I didn't do this by myself. I did it in a core team, mixing leaders from both academia as well as pharma and biotech. And we have a very active LinkedIn page if you want to stay up to date on all the publications from women in the field. Please join our LinkedIn page. We also have free mentorship program, free networking events, and have just this year started offering travel scholarships for early career. So, yes.
Awesome, Danette. Yeah, thank you for highlighting the initiative and for a super, super impressive talk. Really super nice. I think people are very, very curious to see the chemical matter. We had already almost 20 questions in the chat box. So yeah, why don't we just get started chipping away at the questions?
Okay.
I think one of the things, the themes that came out in the questions was the selectivity. Did it come mainly from the protein of interest, the linker, or the E3 ligase choice? Maybe you can, while not giving away the secret sauce, I guess, elaborate on this a little bit.
Can I say all of the above?
Yeah.
We tested so many compounds, and I would say more often than not, you're seeing dual activity, right?
Yeah.
But the chemist could start to pick up on the structural features within the linker, say that we're leaning in that selectivity and really push on the rational design towards that. With CBP, we really, VHL really enables that selectivity. You saw, though, we can, on EP300 side, achieve selectivity with both ligases. Those linkers, yeah, play a huge role, though, in positioning. And I think when you think about that big colon ring ligase complex, EP300 and CBP is 300 Dalton protein. You can imagine, you know, I have just the little bromodomain, which is 10 kilodaltons, but a 300 Dalton, you know, even a slight shift in tilting and docking into that complex could have significant downstream impacts on achieving selective degradation or even steric clashes.
Yeah, absolutely. And yeah, in the as you mentioned, in the EP300 case, you were able to go with both ligases, VHL
Yeah.
and Serblon. So, you know, people are wondering, why did you in the CBB field stay with VHL, or why was VHL chosen as a ligase there?
Yeah, we investigate many ligases beyond VHL and Cereblon even, and we did so even for the CBP project. The VHL base degrader we have for CBP gave us all the features that we wanted. It had that sustained degradation. It had that rapid degradation. And so what you could gain in efficiency by using VHL was very worthwhile for the efficacy that you want, right? And then the big push was how can we improve the delivery and the formulation of this. And so that's where we pivoted to the LAI. But, you know, always in degrader design, you're really looking for the molecule that will be the best molecule for your efficacy. And I think in that mindset, you go in and you just say, we are going to find that best ligase that gets us that best efficacy. And then we will figure out how to make that molecule a drug.
Yeah, yeah, no, totally makes absolute sense. And yeah, I think my soon is wondering, again, you mentioned the turnover rate, I think, of CBP and EP300 as about, you know, 10 hours or so as a half-life of the complex.
Yes.
So how long do you need to sustain the degradation? You showed the 3-week data, which is super impressive with the LAI, and even for a single injection, I mean, it's absolutely
Yes.
incredible data. But you know, how long would you actually need to cover to see an anti-proliferative effect?
You know, typically with most targets in oncology, you have to have excellent target coverage, 85% plus all of the time, right? You really don't want, especially maybe with transcriptional modulators, to allow certain genes to reactivate. So we are targeting for that significant target coverage and keeping the drug at levels that are above that DC90 as long as we can to maintain that sustained degradation.
Yeah, super cool. Yeah, super cool. And I think Junius is wondering, how about neo-substrate degradation? Did you see any IKZF1, IKZF3, GSP1 kind of cell for your your standard
Yeah,
neo-substrates?
so with the Cereblon series, we're continually monitoring this. And that's why global proteomics is so important. But yeah, we haven't, we're not there yet on candidates for the EP300 series, but yeah, this is a super important point. And things like SAL4 are very tissue restricted. So you have to do global proteomics in the right cell background or either monitor it separately with Western blot and in those types of cell lines which express it. But a super important point. But I think, again, I'm not a chemist, but a lot has emerged about understanding how to fine tune that imide handle to
Yeah.
avoid neosubstrate degradation within the context of heterobifunctional.
Awesome, awesome. And then, yeah, with respect to the thrombocytopenia you showed there, there are a few questions here about that. So one question was, you know, you showed the rodent data, and a question was if that also repeats in non-rodents. And then the other question was, can you kind of pen down if that thrombocytopenia is dependent on any of the two individual targets? Like, for example, if you were you know, only seeing that with EP300 degraders or inhibitors, now that you have made the selective version, or only with CBP degraders or inhibitors?
Yeah, so we do not see as soon as we have selective either CBP or EP300, we do not see that impact on megakaryocytes or platelets. So it really looks like it's the dual combination, right, that is leading to it, because we do see it with dual degraders and dual inhibitors as well. We are in the process of doing safety studies in higher order species. I don't have anything that I could share at this moment with that, but it's ongoing.
Yeah, super cool. And I know we're coming up on time, so maybe one last question. And again, there are more than 15 questions still left in the
Okay.
chat, so there was a lot of interest in your talk, Danette.
Okay.
So I guess last question is also the first question in the chat from Christopher Danielson. He says, "Hi, Danette. I was reviewing a dual degrader program for AML and ovarian cancer. I can't disclose the targets as it's a potential first-in-class asset, but he's curious. In your experience, what might be some of the key challenges and opportunities when developing dual degraders for hematologic malignancies and solid tumors, particularly in terms of selectivity, toxicity, and potential resistance mechanisms?" And also, as a add-on question, he's wondering your thoughts on E3 binders versus other binders.
Okay. Wow, a lot to unpack there.
Lots of questions there. So I thought that's a good question to close it up with.
Yeah. Well, look, some of like excellent drugs on even right now in phase later stage phase trials degrade more than one target. The imides are an excellent example. We have IKZF3, 1 and 3 dual degradation. That kind of synergy in those transcription factors, or taking out both of those transcription factors, works very well for efficacy. You know, then the question is, right, in what cases might that be a tolerability issue to have more than one target degradation? But I don't think, you know, depending on the targets, it doesn't always have to be fully selective. You would need that rationale for needing the selectivity, and most likely it is in the tolerability. E3 ligases, I think there's a huge potential out there. The field is still learning about the native biology of many of the E3 ligases. So it's not like even if you had binder, you're guaranteed you have something that tolerates a lot of different non-native substrates, that has high turnover rate and can really compete against new synthesis of proteins once they're degraded. So there's still a lot to learn about other E3 ligases. But yeah, I think this is the next break for the field. Clearly, not just VHL and cereblon are going to work. We see emergence of many decaf substrates. FBXO22 is showing activity in kind of monovalent degraders. So the field will push in this area, and I think more E3 ligases as compared to cereblon and VHL will emerge.
Awesome. Yeah, man, thank you, Danielle, for this really, really impressive talk, and sticking around for answering so many audience questions. So really, really appreciate your time today. And yeah, thank you everybody in the audience for dialing in today, being so engaged today, asking so many questions. We really appreciate that. That's what keeps the conversation going. So with that, I want to again thank Danette for a phenomenal talk. We will be back on May 29th here with Jen Petter, and we will be talking about a structure-based approach of drugging RNA with small molecules. Also a very exciting topic.
Yes,
so
very exciting. Yeah.
So encourage everybody to dial into that as well. And with that, I wish everybody a wonderful rest of their week, and hope to see you soon. All right.
Thank you.
Bye-bye.